Next, we will move forward with the programs that we have for today. And now we're going to go to the program highlights in our first year. This will be a series of pre-recorded presentations from Dr. Gael Pressoir. Is our Haiti quick win lead, coming to us from Quisqueya University. And this will be followed by the presentation from Geoffrey Morris, who is the lead for the trade discovery of the Innovation Lab. And then we will be followed by Elisabeth Garner, who is the lead for the cross-cutting themes in the Innovation Lab. And this will be followed by a live presentation from Holly, who is the Associate Director of the Innovation Lab for Crop Improvement. Thank you.
Hello, everybody. Thank you for hosting us today. My name is Gael Pressoir, Dean of the School of Agriculture at Quisqueya University, and I'm going to be presenting you the work that we've been doing as part of our Quick Win Haiti project. It's meant really as a proof of concept of genomic selection and rapid cycling in the environment of an underfunded breeding program in a developing country. So what's the vision and hypothesis that we're going to be tested? Really, the vision is to develop novel approaches and technologies that will bring an increase in the rate of genetic gain, rapid breeding, especially for contrasting environment, Haiti being made of very various agroecologies of mountainous countries. So with enhanced biotic and abiotic stresses across this environment. And the hypothesis that we really want to be testing during this Quick Win project is first to demonstrate the possibility of enhancing genetic gain with rapid cycling and genomic selection. And second, if the product that we're going to be developing is acceptable, first to me, the breeder, and then to my stakeholders, and especially the growers, but not only them. And so how did it all get started? Our genomic selection program got started really in 2017 through a USAID sorghum millet innovation lab. And our breeding program got initiated in 2013 with a population that we started creating in 2010 with the help of CIRAD from France. And our population was very diverse. It went through a very severe bottleneck through the infestation of Benanaphas sorghum. And it's still pretty diverse because we had the time to do at least 2 intercrossing generations before getting hit by the sorghum infestation and getting at least 1 cycle started. So our population is in light blue. This is the Haitian breeding population related to the East African cow datum. We also have some Kefir and cow datum and Duras inside of it. So really the idea was to do some already rapid cycling even before moving into this project. So we were doing S1 recurrent selection. And the idea is that we were making the crosses during the winter, then evaluating the full SIB or half SIB families, making the crosses within the best, selfing within the best families and the best individuals, and then evaluating the S1 progenies for general combining ability before going back to making the crosses over winter. And so the idea behind genomic selection rapid cycling is that you make possibly 2 or 3 cycles a year by making the crosses between the best individuals' genomic breeding values within the best families. So then you dissociate and then you do the line development and evaluation and eventually retraining, and we'll see why that is important. So what are the results of our first question? And yay, actually genomic selection works. And we're quite happy to have gone all the way to cycle 3 because the result would have been so-so if we had just gone to cycle 2. Now that is good because cycle 3 matches basically is about the same time as closing the loop to a phenotypic recurrent selection on S1 progenies. So here we have cycle 1, not much gain, not significantly different, but actually we kind of went backward. Then we did a little bit better in cycle 2, and we did way, way better in cycle 3. So our marker quality also improved. So the S1 recurrent selection did not do significantly better than our base population, which is already selected individual. It's our training set. But we do have some very interesting line out of it. Now, our best and most promising lines are coming from our genomic selection cycle 3, and one of them from cycle 2. So yeah, so this was an experiment conducted under severe stress, and especially more aphid infestation that we wanted. So that was really, really severe. And these are our yields in grain yield in kilogram per hectare, and they're pretty low. And an interesting experiment. So what are the results? Retraining is absolutely necessary at this stage. Prediction accuracy is pretty good using the new training set, but is awful using the old. Many GS3 lines are greatly overperforming their breeding value predicted with the old training set. Remember, the selection is multi-trait. The prediction accuracy is way better for the lines developed through phenotypic recurrent selection, much better than they are for genomic selection. So we're running out of fuel to be able to do correct prediction. So good results, excellent multi-stress resist tolerance, great rapid cycling recurrent selection. It works, so that's very promising. Now, a little bit too much lodging, so we need to improve our predictive model and our selection index and better integrate some traits. We're running out of prediction accuracy. We're hitting the wall in bricks. We'll see that as our final results. So initial results on physiological breeding is that, that's the Kang index on the left and related to bricks, and with Stay Green as a third trait. So Stay Green is performing great in improving our yield stability. So is bricks, but up to a point. About 14.2, 14.5, adding more stem sugars is decreasing our yield stability, but up to a certain level, bricks is very favorable. So that's the idea, is going to be now combining the physiological prediction with the genomic prediction. So our plan for our final year, at least what's left over of our quick win, validation of our new varieties in participatory trials with the growers, multi-location trials, the validation of our ideotypes, and especially a lot of evaluation of the new training set on more location. Otherwise, we're not going to be able to keep on doing genomic selection. Takes a team to succeed. I thank all my time, especially Jean-Yves Bouchard, but everybody else. A couple of pictures, the people at the Morris Lab and the Butler Lab, and I thank you very much for the chance to present.
Hi, my name is Geoff Morris, and I am Trait Discovery Lead for ILCI. And today I'd like to talk to you about innovation by National Agricultural Research Institutes, why and how. So many national agricultural research institutions have had important successes, but technology transfer from US to NARI has largely underperformed expectations for everyone involved. And for this reason, I am advocating for a greater focus on NARI innovation. That is, NARI conceiving and developing new approaches. And I'd argue that's essential for their success. To foster that, I think we as a community need new habits of mind and program incentives that prioritize NARI innovation over technology transfer. So where do innovative technologies, tools, and methods come from? Well, they mostly come from long process of innovation. So at large seed companies in the US, there is often many-year, many-decade process with collaborations with multiple universities. And that leads to these technologies. It's slow, and it's always really uncertain where it's going to go, or even if it'll lead to something useful. And so when we think about helping NARI, we often focus on how do we do a quick transfer of technology, the products of innovation. And we do that because it seems like it should be fast. It should be more reliable to use proven technology. But I think history has really shown that that's not the case. When we look at the efforts at agricultural development after Green Revolution in sub-Saharan Africa, for instance, high inputs, hybrid breeding, more recently molecular breeding, there's been limited outputs. It's been really, really hard to transfer that technology, and the outputs have really seen limited adoption. So even when they've led to new varieties, it's not clear that those are the varieties that the customers wanted or needed. We've got a whole new set of technologies that everybody's excited about, and maybe they'll fare better, but maybe not. So why not? Why would technology that has done so well in the US not be readily successful in the developing country context? Well, the value of technology is highly context dependent. We've already seen this over recent years in how smallholder practices have been reevaluated. We now realize that there's often really good rationale for smallholder practices, that they're really adaptive to the environmental and social context. And by contrast, we can see now that a lot of top-down agricultural development activity, there was a poor scientific rationale. It was really, in some cases, driven much more by the needs of those that were driving the development schemes. The idea that technology depends on context is really very ubiquitous. It's not just in these smallholder agriculture situations. But I wanted to give an example from smallholder agriculture in the United States, or smallholder, a time when U.S. economy was really based on smallholder agriculture, and this was in the evolution of axe technology from Europe to the U.S. You can see on the right the Anglo-American axe evolved this pole, the heavier bit of metal opposite to the blade that made the axe heavier and more balanced to deal with the larger trees in the US at the time. This is not something that would jump out to us now, right, as a major innovation. But that's the point. It's hard to see innovation if you're not a part of it. It's much easier to see it if you're the one swinging the axe 1,000 times a day for months at a time. And so this is a really reminder to be humble about what innovation looks like. We don't know what it'll look like. And our colleagues at the NARI are in much better place to know what innovation really means. So what process led to successful innovative tools and technologies and methods in the past? One key point is that it was often very intensely focused on customer needs, certainly the case in US seed companies. Further, they use very rigorous criteria for technology development and or adoption at those seed companies that when they developed the technologies that often we're trying to transfer. And so I would summarize that as being goal-directed first and foremost, and then hypothesis-driven. And I note that's in very stark contrast to technology transfer as a concept, because there you really start by insisting on adoption of an existing technology, and then hope that it addresses a customer need. So that's why on the trait discovery area of inquiry, our focus so far has been on really opening up these new habits of mind. Instead of going directly to the specific technological objective, for instance, implementing genomic selection or deploying drones in a program, to really get in the habit of taking a step back, starting with the customer, the end user's needs, and then working step by step to what technological innovations might be possible and what technological innovations might address those needs. And so that is this goal-directed, hypothesis-driven method. And so our focus on this first year has really been getting that conversation going with our NARI colleagues and our US colleagues on starting at that endpoint of customer or end user needs and working back before we even consider what technology might be appropriate, what technology might need to be developed. With that, I'd like to thank especially my NARI partners in the development of the GoHy approach, and I'll be happy to take any questions.
Hi, everyone. My name is Elisabeth Garner. I'm a postdoctoral associate at Cornell University and designated lead for the cross-cutting themes area. I'm very happy to be sharing a reflection on our first year on behalf of our team. First, I'd like to introduce you to our team. As I mentioned, I am from Cornell, along with Munifa Morgan-Bell and Dr. Hale Ann Tufan, both of whom you might recognize from the management entity. And Holly and I are also both part of the priority setting area of inquiry team. In addition, we have Dr. Deborah Rubin, who is co-founder and co-director of the ILCI Project Partner Cultural Practice LLC. And she's joined by Dr. Sarah Eisler, who is a more recent but very welcome addition to our team. We also have Dr. Margaret Mahini, who is Professor of Agricultural Extension and Innovation Studies at Makerere University, Uganda. She is also co-PI of GREAT, the Gender Responsive Researchers Equipped for Agricultural Transformation, along with Holly. In Feed the Future, Innovation Lab for Crop Improvement, cross-cutting themes refers specifically to the inclusion and consideration of gender, youth diversity and inclusion, nutrition, and resilience throughout the research, capacity building, learning, and adapting of all project activities. Further, these themes do not happen in isolation, but rather as part of an integrated system. Therefore, there are opportunities to consider each of these themes throughout the project, and that by doing so, we can more fully and inclusively make a contribution to achieving global food security. For each of the project activities, our aim is to support each COI to address each of these themes throughout their ILCI activities, so that we can achieve our goals and create an inclusive project that mitigates potential harm and maximizes potential benefit. To achieve this objective, we have several ways that we are available to work with COIs and support ILCI activities. Engagement and capacity building are how we will increase awareness of and attention to each of the themes. In addition to reaching out to each COI individually, if there is interest, we will also support a community of practice among COI cross-cutting theme team members. We also have 3 types of trainings planned over the next 3 years, building from the sensitization or introduction of the concepts to introducing tools to use in the field. Thirdly, we are available to support the COIs in their research activities by providing resources and feedback on everything from research, design, tool development, to analysis. For our work from this year, we have some points to highlight. For capacity building in the first half of the year, the work was focused on preparing for the project. At the January kickoff meeting in San Diego, the cross-cutting team introduced the value of addressing gender and youth to achieve social inclusion with the 2 trainings, social inclusion and implicit bias. The inception meeting has been followed up by 2 sessions organized by the management entity, including a recent workshop on systemic racism and international development, led by Ben and Lauren Reese. The past year, the team has also provided guidance on addressing the cross-cutting themes in the RFAs for the Center of Innovation, reviewed the COI letters of intent and the full proposals, as well as participated in the co-creation phase to support the full application development. The second half of the year has been focused on the first training we will share as a team, an online sensitization training. These trainings emerged in response to the COVID-19 restrictions and the following uncertainty. While in general, we believe that the strongest impact on these themes comes from in-person and in-depth guided personal reflection, particularly on the topics of diversity and inclusion, we wanted to still provide the COIs and the ILCI team more broadly with some sensitization to each of the cross-cutting themes. We are currently in the process of designing 4 topic modules for each cross-cutting theme. Each one is designed to be delivered asynchronously and will be accessible in terms of language, so subtitles and translated, as well as technology, so you can download them and participate in them offline. The objective is that each participant will walk away with an understanding of the basic terms and issues for each theme, understanding the importance of including that topic throughout your programming, along with examples of how it has been integrated successfully or not so successfully in the past. And while they are not guided lessons, there are still points for self-reflection and a reflection on one's own institution. Likewise, our research activities for year 1 pivoted to work that could be done remotely and before the COIs were identified. This research has also served to directly inform our capacity building efforts as it sets up a deep dive into ILCI and COI's capacities and needs around the cross-cutting themes at the institutional, programmatic, and the individual levels. This work was led by our summer graduate assistant, Emily Hillenbrand, who is a PhD student in global development with lots of experience in the field. This work has 2 major outputs. The first was a literature review to examine existing frameworks for capacity review. Emily's review found that there was great resources that, however, they tended to be focused on either gender mainstreaming across institutions or evaluation of projects along a continuum, and were very specific to the organization that developed them. However, it was difficult to find a framework that worked for projects like ours, where projects were embedded within institutions or multiple institutions, and with multidisciplinary teams who might not have the experience or the background to begin with in terms of cross-cutting themes, particularly gender. The second part was a document review of ILCI, or sort of a pilot test of the framework, but also an opportunity for self-reflection. You may have received an email from her requesting your CV this summer as part of this project. In looking at the team, she found that the planned activities offered a lot of opportunity for increasing understanding across the ILCI technical team. However, most of the social science or non-planned science degrees were concentrated into a few areas of inquiry. She warned us that this could create the potential to silo the team and these topics into the particular areas where they are. In addition, a review of the budget finds that only 5% of the total budget is dedicated for all cross-cutting issues. This is actually below the CGI recommended standard. The goal is 10%. However, there may be opportunities to recalculate this now that the COI activities are proposed and as we get going with the project. So reflecting on the challenges for year 1, our major points were of adaptation, particularly to COVID, as I'm sure for many of the teams and COIs can express. In response, we shifted to an online program, as I mentioned. However, there have been challenges in planning for the future, which has also been a major activity of our group. We also have started to make more progress on this now that the COIs have been announced. Welcome COIs. And we know the regions and focus of the programs we will be working with. However, we have spent some time outlining roles and responsibilities as activities shifted, as did our team composition, including the addition of myself. We also had to adjust timelines and expectations. One thing we did not talk enough about, and we do not talk enough about, is just the extreme stress and uncertainty that COVID has caused and the extra time that we all need to make these adjustments in our work. But we also found that our original timelines needed to shift also in response just to our optimism in how fast some of the larger paperwork could be processed in some of the bureaucracies. This also caused some confusion in the budgeting process, which also responds back to the importance of defining activities as a team and then among team members, particularly working with outside Cornell and with partners. We also look forward to working together to better understand the reporting process. As our team shifted, so did how we report, causing a bit of confusion at the end of this year, but we already see how that things can be smoothed moving forward. And we also look forward to more communication between AOIs and the management entity as decisions are made and the program grows. As we move forward into the next year, we will be reaching out to COIs in the very, very near future if you haven't received an email already, and to start discussions on long-term engagement and support. And we've already initiated planning for next year's training to build from the online sensitization trainings. On behalf of the Cross-Cutting Themes team, we look forward to working with all of you as the project continues. Thank you for letting me share, and enjoy the annual meeting.
Good morning, good afternoon, and good evening, everyone. I'm Hale Ann Tufan. I'm the Associate Director for ILCI, and today I'm going to be talking about giving you some behind the scenes on the process that we followed to identify and select the ILCI Centers of Innovation. As you know, the ILCI vision really pivots on serving as a support system for NARIs on their journey to self-reliance for crop improvement. As we wrote our proposal and started our program, the key question we kept coming back to was how. How do we effectively serve as a support system? And as a thought of how to structure the support system, we didn't start with a process, but really started with principles. So we started thinking, what are the pillars on which we could, the foundations on which we could place an RFA and a process of granting that would really uphold that vision? And here's some of the things that we came up with. The first of all is that we would like to aim to empower NARIs to pave their own path towards sustainable, climate-smart, equitable, and effective crop improvement programs. And we really see NARIs as being in the driving seat of the quote-unquote modernization agenda that we're seeing currently around crop improvement globally, so that it fits their impact pathways and resource base. We also see a process of granting as being one of design and selection processes that are co-created and co-visioned, so that this was a central principle to everything that we did. And we decided we would be open to ideas and approaches and offer support from ILCI through area of inquiry leads and cross-cutting teams. We spent the first 4 or 5 months of the grant last fall thinking through an RFA process that would uphold these values and the structure. And we put out a call about mid-February, and I'll go through the details of the call and how we processed each step of that. The first is eligibility. So in the RFA, we had these eligibility requirements. Being a 5-year project and spending a year getting started, we set a duration of 3 years for the Centers of Innovation. But we didn't see this as an end at the end of the 3 years. What we really would like to see is contribution to building a long-term, sustainable, self-driven, and effective breeding program. So that was one of the criteria that we were looking for reviewing the applications. We see this as a seeding towards a longer-term sustainability. We also had mandate crops. So we looked for product profiles that were focusing on one of the mandate crops that the ILCI covers. A major requirement was that the applicant PI must be within a NARI in one of the Feed the Future eligible countries. And as Elisabeth just presented, we really place high importance on cross-cutting issues, and we wanted to make sure that the development goals were really clearly indicating how becoming a COI would increase benefits for women, youth, climate resilience, and nutrition within the NARI countries. And we set a budget maximum of $1 million total for all 3 years and all partners involved. In the RFA, we also set out priority criteria. Linkages with US institutions, especially with MSIs, was a priority selection. We also wanted to make sure that applicant PIs, applications were led by minority groups, women, and early career researchers as a priority. Regional partnerships with other NARIs, universities, institutions, and CG centers was important. And since the work that we do is multidisciplinary, we look for proposals submitted by an interdisciplinary group of co-PIs. In the RFA, we asked to have capacity building plans thought out, articulated at the individual as well as the institutional level. The graduate student training plan should be in place both for long-term degree granting, but also short-term through workshops. And for each of these, we asked that the strategy ensured equal opportunities for underrepresented trainees in place for all programs planned. Cross-cutting themes was clearly articulated as a priority areas. We asked that product profiles should really seek to address gender and youth-based constraints, show potential for economic inclusion for women and youth, and include traits that address malnutrition and increased resilience. We were looking in review at applications that designated a team member with a relevant background and expertise in gender and youth inclusion, nutrition, and/or resilience, and that there was adequate funding allocated to really do research on the proposed impacts of the TTMs that were adopted on gender, youth inclusion, nutrition, and resilience. The RFA process was multi-staged. We started with a written questions period as well as a webinar that we hosted. We went to a letter of intent stage and then went through co-creation and final proposal submission with a short list of applications, finally granting in September this year. For the written questions and webinar phase, we had 150 registrants from 41 countries who showed interest in this first stage. When we got to the letter of intent stage, we had 100 applications from 26 countries, and we moved forward with a short list of 10 applications from 5 countries through the co-creation and final proposal submission stages, ending with 4 centers of innovation you'll hear a lot more of later this morning. So I'll go through each of these stages. For the written questions and webinar, we posted the webinar online, the video, and we also collected all questions we received beforehand, during, and after the webinar so that each applicant had full access to all the information possible before they submitted. At the letter of intent evaluation, we narrowed the list of 140 applicants the management entity narrowed based on criteria of eligibility to a long list of 40, which were then reviewed by 10 reviewers to categorize into yes, no, or maybe categories. And we used evaluation criteria to cover the basic tenets that we were looking for. The first was the potential for impact from released varieties that met the priority product profiles articulated in the proposal. And how, as areas that were identified as weak, as they were strengthened through this investment, really would help achieve that articulated impact. We looked for a clarity of plan for including gender, youth, resilience, and nutrition into proposed work, a vision for capacity development, as well as the capacity of the institution, partners in the region, and internationally. All of the feedback we received was consolidated and delivered to the shortlisted applicants as action points to think about through co-creation, and to unsuccessful applicants as reason why they were not selected. During the co-creation phase, we sent an initial letter to all shortlisted applicants because we saw areas that were consistently weak across the shortlisted applications. One was differentiating their current application work from previous and other work. So we really wanted to not be redundant with ongoing projects or past projects. We also saw that many times in applications, the financial contributions of the host NARI institutions were not always clear. So we asked to see details of financial contributions because we believed having the host institution having skin in the game and really being together with us in this co-creation process was really important to sustainability. We also looked for clarity on cross-cutting goals, staffing, and budget, as we saw this was not always clear in all applications, so we prioritized this in the co-creation process. We then set about organizing co-creation calls. The first calls were with the management entity to inform all shortlisted applicants on process and give them directly the feedback from the review process. We then organized a matrix of calls with each applicant an area of inquiry and cross-cutting leads so that they had the ability to consult and get any feedback or further information they felt they needed for the final proposal. We made sure that the applicant PI was responsible for documenting and acting on all the points raised during the co-creation process. We then came to the final proposal stage. We received all 10 final proposals from the shortlisted applicants, and these were reviewed by 20 reviewers. So we almost had double the amount of reviewers as applications. But I think that was important because we intentionally selected reviewers for diversity of backgrounds and experience, and that was very intentional. Because we wanted to get different views and reviews and have discussions and further thought to consolidate. Because as we know, the more reviews and diversity of views that you get, the better the chance of selecting good proposals. The final selection was strategic to balance quality of proposals, but also regional applications and crop coverage. This is just a snapshot of the review process that we used in PyStar. We had qualitative feedback section where each reviewer could enter their thoughts on each application, but they also had a quantitative 5-part rubric where each application could be scored based on the same evaluation criteria we had in the LOIs, but here they could individually score them. Lastly, we asked each reviewer to recommend and rank the proposals. So they would give us their top 5 proposals in rank. So we could use that in evaluation. I'd like to spend the last 2 slides reflecting on what we learned and how we would adapt this process should we go forward with another RFA round. Managing the applicant pool was a big part of what we did. We were admittedly weak in reaching out to and including MSIs as US partners. And we want to rectify this going forward because, especially with the discussions we recently had with the Innovation Lab Councils of Directors, that we really need a more intentional, targeted strategy to include MSIs in all of our work. We also had too few women PIs, and we should have rectified that going towards long and short listing processes, so we were prioritizing women PIs. So that was something that would change going forward. We also had multiple applications per institution, which did end up being repetitive at times. So going forward, we would limit to 1 application per institution. We had 2 instances of mergers where we had between the long list and the short list, we discussed with applicants who were either from the same institutions or had really alignment in their work to consider mergers between their applications. But that process needed further thought. In one instance, it worked out really well with Gael and Roberto, who you'll hear from later today, but in some instances it did not. But the bottom line was that having this applicant pool showed us that we have a great number of potential partners to work with, but supporting them going forward and keeping those linkages will require good communication and responsiveness on our part. The second thing we were really careful about was trying to avoid confirmation bias, because we know there are very good applicants who already have many projects and who may be very good at applying to this kind of opportunity. But we wanted to make sure that we weren't always selecting the same individuals in the NARIs that already had many grants. So we had a transparent reflection process at each stage. We were having discussions with the review teams and also amongst ourselves to make sure and checking ourselves, being self-aware about our tendency perhaps to prioritize really good applications that may have a lot of redundancy with ongoing work. We also had very diverse reviewers, and we used those reviews and balancing everyone's views and selecting the final applicants. We tried to balance potential with established reputation in selection, and we checked the scope of work and vision were not overlapping with ongoing work from the applicants. And the last part is the co-creation selection process. It at times was hard to consolidate the diversity of views and reviewers, and we did spend considerable time negotiating and selecting in the short and long list processes, but it was definitely worth it. It was important to meet the applicants where they are at times, especially true for cross-cutting issues, as now we focus on potential for longitudinal change and working with our finalists to build the vision, especially for cross-cutting issue integration into their work. And what we did see was that applicants who were responsive, really communicated well, and integrated everything during the co-creation process was a good indicator for success. And speaking of success, I just want to say that we're extremely excited and privileged to be working with these 4 centers of innovation. We'll hear a lot more about them very shortly, but I just want to thank you all for joining us this morning and joining me in congratulating our 4 centers of innovation in their future success. Thank you.
Thank you so much to all the presenters, Gael, Jeff, Elisabeth, and Holly. At this time, I'm going to welcome the presenters to come on stage to turn on your video and your audio as I open the session to questions from the audience. I'm going to tell our audience members to please raise your hands and you'll be picked to ask questions. Unmute yourself, turn on your video and ask your questions after you're called upon to ask the question. And if you're having trouble locating where your raise hand button is, feel free to put in the chat box that you have a question and you're going to be called upon. So while we're waiting for questions from the audience, I have a question for Gael. Gael, you had a very great success in the past year, and I'm wondering, as you transition from Quisqueya to the Center of Innovation together with Roberto, what would you have done differently? And what do you think ILCI could have done better? Please turn on your video.
I did turn on the video. I don't know why I'm not seeing it. Video problems. Yeah, I don't know. So video should be on. Select the camera. Okay, so we'll just talk. I don't know why the video is not working.
All right, you could just go ahead and answer the question.
Okay, so yes, thank you for your question. So what would we have done differently? I'm not quite sure right now. I would definitely do a lot of things differently in terms of the breeding process, but that I would have to go back 3 years from now and learn, because it's all about monitoring also and learning from your mistakes. And one of the things, we didn't have much variation to start with. We had done strong selection initially for lodging. So I was expecting some lodging in my GS population, but not as much as I saw. So that's one thing that's critical to establishing a genomic selection rapid cycling. It's because when you're doing the rapid cycling without doing the phenotypic assessment, but just doing your predictive models, every trait that is not in the model, sometimes it can go haywire, and that's something that we did see for lodging. So going back in our breeding, we would definitely change our prediction model and our selection index to account more for lodging and better phenotype lodging in our training set. So these are the type of things that, as you're learning to use a new technology, you're also learning about what traits to include in it or not, and what goes wrong. So this is not about so much what we would have done differently. It's much about learning from the process and getting better at doing it. Otherwise, yes, with CACSHA, we're going to be doing more, expanding, transferring some of the technology south-south. So from our colleagues in Costa Rica, we're going to learn to be better at participatory evaluation and accounting for growers' need, product profiles, because they have more experience than we do. And in terms of novel technologies such as physiological breeding and genomic selection, we're going to transfer it the other way around. So it's going to be an opportunity to learn and expand on the work we've been doing, and doing it better, I guess.
Thank you so much, Gael. I appreciate that you are learning from the past and making your program better. Now I'm going to move on to Mark Cooper. He has his hands raised, and please ask your question. Unmute yourself and please ask your question. Thank you.
Hi, thanks for the opportunity to ask a question. I sort of have a couple, but I'll only ask one at the moment, and I'll actually, maybe it's a question or a discussion point for Hale. I think that it was interesting, the description of the process you went through to help people with the application process. I mean, reflecting on this and thinking about the actual evaluation of the process, I think going forward, probably we need to think a little bit more effectively about how to help the reviewers to actually conduct an effective review I think there was sort of an imbalance between the amount of attention that went into helping the reviewers do a really good review versus the amount of effort that went in to enable people to put in really good applications that were aligned with the objectives of ILCI. And I think that making the assumption of relying on diversity to get good reviews is sort of a safe one, but I mean, I always remind myself that 10 random numbers isn't much better than 100 random numbers if we don't really know what we're actually doing an evaluation for. I'm not saying it was as bad as that. I was just trying to dramatize the point that I think ensuring we do good reviews that is aligned is probably something to think about to really improve the effectiveness and balance up, ensuring we get the best out of all of the effort that everybody's actually putting in. So it's a sort of comment and sort of engaging in that discussion. I mean, I don't like to believe that the reviewers have some golden wisdom that they're using to sort these things out. It's pretty messy on both sides.
Yeah, definitely. And we saw that not so much in the quantitative rubric, but in the qualitative comments that really came through. And we saw that everybody was interpreting things a little bit differently, which in the end we ended up embracing because we had very different views. We had people who had really deep knowledge in certain regions and were giving us really in-depth feedback on the potential for that applicant, which we thought, this is great, right? That you wouldn't get that if you didn't have that person in place. So I agree, it wasn't easy, and having 20 reviewers in itself was difficult. But at the end of the day, it was a balancing act, and I think it balanced out well. But going forward, maybe instructions and more help to the reviewers as well noted. Thank you.
Thank you very much for that question, Mark. We have a question in the chat box from Dave. And this question is directed towards Gael. It says that you said that the study was done under significant water stress and insect pressure. Can you talk a little bit more about the level of quality of the phenotypic data from the studies that was used to show success of rapid recycling? I'm going to take
yeah, and maybe I can just explain the question a little bit. I had trouble figuring out how to raise my hand. And so, whenever you hear the words, the study was done underneath significant stress or insect pressure, immediately I start to worry about the quality of the phenotypic data that you're trying to measure any sort of gain from a genomic study. So just help me understand, what was the quality of that phenotypic data coming from these studies?
Thank you, David. So yeah, I'll answer that. So the irritability levels are quite high. Actually, whenever we put a lot of stress on, usually we get higher irritability because you get higher variation of response of the phenotypes. So we did check the quality of our data through repeatability and irritability. So of course we have some levels of variance, and that's why it was a complete experiment replicated 4 times also to balance and to be able to measure both the broad sense and then genomic irritabilities. And so they're pretty high overall of that new training set because it's an advantage to basically start the retraining. Now, the reason why we had a little bit more stress than we were envisioning, it was Corona time at the times the plant were growing. So there might have been a little bit less presence in the field. We were still struggling on how to manage our field situation, everything like that. And especially the spring season was a knockout. We had a very, there was no rain the whole season. And the aphid just loved no rains. So we did water a little less than we were. So it was not meant to be as much of a stress. We just had some climatic, one of our worst spring season that we've ever had in the past, I think 20 years. It's the worst spring season that we've had. So it was an aphid blast. And we decided not to treat because aphid is one of our target of selection, and we decided to have it, except that I just didn't know they would have such a feast. But it was an interesting experiment because the data shows to be quite consistent, highly irritable. Since we're breeding for stress environment, I definitely need an additional data for that training set under expressing their yield potential. But I have no regrets that we evaluated this population under this level of stress, because the data is heritable for most of the traits, and it's a nice experiment in terms of showing that our genomic selection. I'm not sure we'll see as much of a difference for the optimal yield, but that is yet to be seen. But we will run that experiment, and actually, that thanks to CACSA, that experiment will be run not just in Haiti, but also in Nicaragua, and in Haiti in at least 8 locations. So you'll get to see better data on that new population.
Excellent, thanks.
Thank you so much. I'm wondering if there are any questions for Jeff or Elisabeth before we go to take a quick break.
So maybe I've got another one if it's okay. I put my hand up, but
all right,
pushing
go for it.
Is that okay, Sarah?
It's okay.
One of the things that I do, it sort of cuts across to Jeff, but I'd actually congratulate Gael on his willingness to show some things that didn't work. And I think that that's really important for the success of an innovation lab. I think that, you know, really you're going to learn a lot from the difficulties of putting some of these technologies in place. There's sort of often this expectation that something like genomic selection is almost bulletproof, but if you've lived and died by it, you'll find you'll mostly die by it if you don't actually get things pointed in the right direction. And I think Gail's results are a really good learning opportunity for everybody, that high heritability tells you you've got some separation. It doesn't necessarily tell you where you're pointed at. And I think that you sort of, that variability you see across these cycles of selection is sort of pretty informative. And I sort of saw some comments a bit from Ed Buckler, and I think your point, Gail, about getting the sort of environmental situation right to actually create a training data set you can have some confidence in is sort of something that really needs some really deep thought, because I think we can always rush into pushing that technology just because we have to hit some timelines. But I mean, sometimes not doing something might actually be a better decision sometimes if you just haven't got quite the right alignment. And I think Jeff sort of made this point about the difficulty of transferring from technologies that should work to putting them in place and getting them to work for you. It's sort of a real learning process.
Thank you so much, Mark, for that comment. And I want to thank everyone who have just presented and asked questions. And I apologize that we are running 5 minutes late, and I'm going to release us for a 10-minute break. Please let us be back at 9:30 in 10 minutes. Thank you.